Today, I'm going to spend some time talking about new promising therapies and updates in research in HD. Let's see. There we go. All right, so an outline of this morning's talk, I'm going to spend a few minutes talking about neurologic music therapy that's being done at the University of Colorado, and then we're going to spend some time talking about gene therapies. Can you hear me okay? Okay. So we're going to start with just a review of what gene therapies are, how they work, and then review trials that are being done with gene therapy in HD. So there's an entire field of study that looks at the effects of music on the brain. There's a CU specialist, Dr. Isabelle Huard, who is studying how neurologic music therapy, including auditory cueing, affects things like fine motor control in people with HD. And so her study is looking at a 15-session neurologic music therapy intervention. So what it entails is there's a first visit where we do some testing. So a lot of that is things that you're used to are things that you're used to, like the UHDRS. We do different surveys and perform a brain scan. And then there are 15 music therapy interventions. And those can be done virtually in the home, or the participant can come into the clinic. And then at the end, there's a repeat where we do post-intervention testing with the same surveys. Surveys include the UHDRS, a grouped PEG test. So that's one where we have someone do like a fine manual dexterity test, and then different mood assessments to check for things like anxiety or depression. At the same time, we're looking at a specialized brain scan called a magnetoencephalography, or MEG for short. That is what the image on the right shows. So we have a few participants who've undertaken this before and after, and we've seen some possibility that there might be a signal change in the magnetic fields in the motor areas, but we really can't make any type of conclusion from this. We need more participants to be able to tell if there's any meaningful change. So if anyone's interested, all that's required is an HD diagnosis in someone that's between the age of 30 to 85 and has been on stable medications for the past month. They need to be able to move fingers and hands and not have any metal implants that would interfere with the study, but that's not all metal implants. We would like the exclusion criteria to include dementia, so people cannot have dementia or be engaged in other research studies. It's a little dependent on what the other research study is. So if anyone is interested, we'll give you our contact information at the end of this presentation, or you can talk to me or Nicole at the back during the morning. So let's move on to gene therapies in HD. So gene therapy is a technique that uses genetic material to treat or prevent a disease. So in order to understand gene therapy, we're going to spend a few minutes reviewing the basics of genetics and the basis of genetics in HD. So genetics 101, all our cells have a copy of our complete genome. So all of the instructions and blueprints for making all the proteins in our body come from our genome. And so all our cells have a copy, and it's tightly wound up into chromosomes. We get one chromosome from our mothers and one from our father. The DNA itself is made up of 4 different chemicals, which you see on the picture on the bottom left. And when a protein is needed in a cell, that piece of DNA is called up and is transcribed into RNA, which is a messenger that takes the message from the nucleus where the DNA is housed into the cytoplasm where the machinery to make a protein is, and it makes that protein. And the protein can then go on to perform the cellular functions. So let's spend a minute talking about the genetics of HD. I'm sure many of you are very familiar with this. But every case of Huntington's disease is caused by the same thing. So there's a genetic variance where there's an expansion of CAG. So those are 3 of the chemicals that make up. So most people have up to 26, and that's perfectly normal. And then up to 35 even doesn't cause disease. But beyond that, the increased CAG repeat will cause Huntington's because that repeat expansion in the DNA becomes a repeat expansion in the RNA, which goes to make a protein that is particularly long. So you can see on the top in the normal length protein, those red, that's where the CAG repeat is. That's where the corresponding area is in the protein. On the bottom is Huntington's disease protein. So that is a much longer area. And so when the protein is long, it gets very sticky and it clumps up, aggregating in the cells. When it aggregates in our brain cells or our neurons, it really wreaks havoc on the normal cellular function. And so what I equate this to in clinic is someone who has, for example, like a messy house. So when our houses get messy, we can trip over things or lose things, or there might be food somewhere. And it's nice because we can just clean that up. But our cells don't have a really great way of cleaning up all this extra HD protein. And so the signal that the cell gives to itself is to die. And so these cells end up dying. So if it happens to one cell in the 3 billion cells that we have, it typically is not a big deal. But because this protein is accumulating in all of these cells in the brain, all these cells end up unfortunately dying off, and that's what causes the symptoms of HD. Typically, this occurs first and foremost in a deep area of the brain called the striatum, which you can see on the image on the right. So the DNA to protein image that we see here is a little bit simplified, and it goes through a few extra steps. So in order to understand the gene therapies that we're going to talk about today, we're just going to spend a moment talking about those extra steps. So it's a very complex picture, so we're going to break it down piece by piece. But overall, what we see here is the DNA in the nucleus on the left gets transcribed to messenger RNA, sent out into the cytoplasm, and then turned into a protein. So let's just look at the very first part of this. So starting in the beginning, in our DNA, what are the gene therapies that are available? And so right now, we don't have clinical trials for any gene therapies that act in our DNA, but there are a few technologies which enable us to do that. So I'm going to briefly mention them because I do get asked about them from time to time. So the first is a zinc finger protein, which you can see in the pink box on the right. So that's the ZFP. So that's kind of a editing out of the CAGP. There is a preclinical trial looking at the zinc finger protein in the lab, but we're not going to go into that today because it's not really gone to humans yet. The other one that I get asked about more commonly is CRISPR. So CRISPR is a newer, very popular technology that's being used a lot in the lab and has a lot of promise. So even though there are no clinical trials with CRISPR, it has already been helpful for people with HD because it has enabled better animal models and better preclinical data. And there are several centers that are looking into CRISPR for clinical trials for HD, but we're just not there yet. Okay, so we're going to move on and talk about once the DNA has become RNA. So DNA becomes RNA, and then it moves out into the cytosol to become a protein. So in its RNA form, it's very vulnerable. And remember, our goal is to basically get rid of that last step so it never becomes a protein. And we can use the vulnerability of the RNA to target it in 3 different ways that we're going to talk about today. So the first way we're going to talk about is an ASO, or an antisense nucleotide, or an antisense oligonucleotide. So I'm going to show you another picture of basically what this is that's a little bit simplified, just so we can talk more about the ASO. So, like we talked about, the piece of DNA is needed to make a protein. It gets called up, turned into a piece of RNA, sent out, and made into a protein. That mRNA, the messenger RNA, is very vulnerable. And it's a great spot to target to prevent it from becoming a protein. What we can do is we can create a little strand of DNA, so some nucleic acids that correspond to that RNA, send it into the cell, and when those two bind, the cell recognizes that piece of mRNA bound to the antisense oligonucleotide as abnormal. And the cell's own machinery will destroy that, break it down, and prevent it from ever becoming a protein. And so that's the way that ASOs work. In general, there are 2 types of ASOs. There are ASOs which target only the abnormal mRNA, and so only the abnormal Huntington protein will be prevented from being made. There are also ASOs which indiscriminately target the mRNA for the Huntington, the mutant Huntington and the non-mutant Huntington, so that no protein gets made, whether it's a normal Huntington protein or the mutated Huntington protein. So the first medication we're going to talk about is a trial for a medication called tominersen. So tominersen is an antisense oligonucleotide designed to lower the amount of mutant Huntington protein in the brain. It's administered via spinal tap. So you can see that on the image on the bottom right. So basically, take a needle, put it in the lower part of the spine where the cerebrospinal fluid is, inject the tominersen, and then it gets basically distributed throughout the CNS. the central nervous system. And so that tominersen can go throughout there by just putting it right in the lower spine. So this one has been around for a while, and there was really great data from very early trials that found that there was Huntington lowering ability. However, a few years ago, when they looked at this in humans over a longer period of time, they found that higher dose tominersen, or more frequently dosed tominersen, actually had people do a little bit worse. And so you can see that on the left. So what we see there, here. So this is the baseline, how people came in. And the gray lines and dots are the placebo. So that's just basically a sugar pill. The orange one is people who were dosed every 16 weeks. And the blue line is the people who were dosed every 8 weeks. And you can see that the further down that they go on the right, the worse that they get. So the blue line is actually doing worse than the people who got the placebo. And so once this data was seen, the trial was stopped. And so it was March 2021. However, analysis of specific groups within the study suggested that lower dose tominersen may benefit younger people with earlier onset disease. And so that's what led to the Generation HD2 trial. So the Generation HD2 trial is being done in a younger group, so that's 25 to 50 years old, with prodromal or early stage HD. And I think it's important to note that this is a study that's being done for people who have prodromal disease, so people who don't necessarily have motor onset yet. The goals of it are to determine side effects, changes in the amount of mutant Huntington protein in the spinal fluid, changes in imaging, and then changes in function. So for example, being able to move, think, or perform daily activities. This is at the very start. The expected end date right now is not till 2027, but it is one that is recruiting. All right, so let's move on to our other type of ASO. This is the one that targets either the normal or the mutated Huntington. The next one we're going to talk about selectively targets just the mutated Huntington. So this one is a pretty clever setup. So it uses some variation in the mutant Huntington RNA code to only target the mutated version of the allele. And that way, the healthy version can go on to create healthy Huntington protein. This was also a study that was done in earlier phases. So this was the Precision HD study. The Precision HD study a few years ago did not demonstrate that it was able to actually lower mutated Huntington. So that was also stopped in March 2021. But the company that sponsored that one went on to develop a third type of ASO, and that's what's being tested now. So it's mutant Huntington specific. It's a phase 1/2 trial. Unfortunately, it's only being done outside the USA. So it's being done in Europe, Canada, and Australia, but it's one to monitor. They have 36 participants. It's administered via lumbar puncture, and they're looking at safety, tolerability, and early results show that it is actually safe and well tolerated, and it's decreasing the amount of Huntington by more than the original trials did. So this one's, the most recent data showed that they observed about a 35% decrease. So we're going to move on to talk about the next type of therapy that targets that mRNA before it can be made into a protein. All right, so we're going to talk about these small molecules. And under the small molecules, you can see what says splicing. And so what does that mean? So when the mRNA is made, it can be put together in different ways. And so the small molecule we're going to talk about basically changes the way that it's formed to prevent it from becoming a protein. And so the way that it does that is you have 2 exons, meaning 2 areas that are expressed right next to each other. And that basically provides instructions to make a protein. What this molecule does is it inserts an extra piece of information between those 2 to say, stop, don't continue this, and break it down. So that piece of mRNA gets destroyed. So this is an oral splice modulator called PTC518. So really exciting, that's an oral medication that it's not an injection into the lower back or into the brain, which we're going to talk about more in a few minutes. It reduces the level of Huntington in different animals and lab models of Huntington's disease, and it's a phase 2 clinical trial in people with very early symptoms of HD. All right, so we're going to go over a little bit of the data that's come out so far for this. So we know that this actually does decrease the amount of Huntington RNA. So what you're seeing on this graph on the top, that it says that just because that's how the dosing is. So don't worry about that. The gray line is the placebo. So you should have about 100% of our normal RNA if we're doing nothing. And that's what a placebo is, the sugar pill. It doesn't really change much, which is what we would expect. And then after we give higher doses of the PTC, the amount of Huntington RNA decreases. This is in healthy volunteers and it's in the blood, not the brain, but it's a really good sign. This is over 24 hours. So the next step was looking at how long does this effect persist? And so what you can see here is the gray line on top, that's the placebo. So someone will take the placebo and nothing really changes about their RNA, which is what we would expect. But when they take the drug, their mRNA levels of Huntington protein goes down and it stays down for a while. It doesn't get back up for a couple of days. This is also in whole blood. So you might be wondering what's going on in the brain. So they looked at the amount of free drug that's available in the CSF, and they found that as it increases in your blood, it increases in your brain. So it's able to get into the brain. So another good sign. So initially this trial is for people with very early Huntington's, but then it expanded to include people with movement symptoms and mild difficulty with their daily tasks. And they also extended the trial from 3 to 12 months. So those are promising signs. Whenever a trial like this is changed, the FDA has to go back and take a look and make sure they're okay with the new protocol. So the FDA at this point is still thinking about this. So it's on hold in the United States, but it is ongoing in Europe and Australia, and hopefully will restart here soon. Who can participate? People above the age of 25, people with 40 to 50 CAG repeats, and those who are at or near their normal baseline functional capacity and independence scale. All right, the last type of intervention we're going to talk about to prevent Huntington protein from being made are basically a viral intervention where a virus containing helper genes is sent into the body and prevents Huntington protein from being made. So this is pretty exciting. So we're going to spend a few minutes on this one. So the AMT-130 is a gene therapy delivered directly into the striatum. So that's the area that's affected first and foremost in Huntington's disease. And the goal is to slow disease progression by interfering with the production of the protein. It's delivered by a one-time neurosurgical procedure. So in order for this protein to access the area it needs to have two steps. One, it needs to get into this very deep area of the brain that's about where, if my fingers would intersect, where you would find it. So it's hard to get there. And then it also has to get into the cell. And so we're going to talk about how we get both into the cell and into the deep part of the brain. So in order to understand this, we're going to spend a minute just talking about viruses. So viruses are able to infect us because they have spikes on their exterior shell that trick the cell into letting them in. And then typically an active virus can essentially hijack the cell's own machinery and endlessly create more viral particles. Scientists have developed a way to program a virus to use our cell's machinery to make therapeutic medication. There's a major focus on this in research right now, not just in Huntington's, but for basically across the board. It's a very exciting and promising area, but it still has some work to do. The way it works is we select a virus that does not cause illness in people. So we're much more familiar with viruses that cause illness in humans, but we interact with viruses all the time that we don't know about because they basically don't infect humans. So we pick one that does not infect humans. We keep the coding so that the virus can actually get in the cell, and then we take out all the viral genetic material. So it's basically an empty shell, and that way we can put in what we want. So instead of having virus genes, we put in specially engineered genes, which are made to block the production of the harmful Huntington protein. So a company called Unicure has done that. They have a compound called the AMT-130. It's made up of the virus shell, and the shell is from an adeno-associated virus. Most of you probably haven't heard of that because it's just not one that really infects people. So it's not something that's really big in the news. But it is used a lot in research. The adeno-associated virus family has many different types. They've used type 5, but there are other types being used in different types of research. The AAV, as we call it, has been modified so the interior has a microRNA, so a little piece of RNA that is a helpful piece of genetic material, and it provides instructions to decrease the production of the abnormal Huntington protein. And so once the neuron has been treated with the AMT-130, it actually can continuously make its own Huntington lowering molecule. So overall, this should result in reduced production of Huntington protein. And the goal is that once this is in, it will last for a very long time, possibly even for life. So that's how we get the molecule into the cell. But how do we get it into the brain? It is a one-time neurosurgical procedure that aims to reduce Huntington protein. So we have to drill a hole right in the skull with a microcatheter that gets sent in, and then the molecule is infused over several hours. So the goal of the study, the primary goal is safety. So again, the AAVs or adeno-associated viral vector therapies are newer. And so we really have to make sure that this is a safe therapy. We don't know how long this sticks around, and we don't have the years or decades of experience with using this that we do with other types of therapies. So safety is the number one concern. Tolerability is the other, making sure that the drug and the procedure are both tolerable to our patients. Understanding how long the compound sticks around for, so we absolutely don't know how long it's going to last for. And then exploratory endpoints is, does this affect progression? So that's really ultimately down the road will be the goal of understanding it, but it's in early phases. So this is something that's more exploratory at this point. The timeline for this, so there are 10 patients in a low dose cohort. So some of them were treated and some of them were given placebo. In a neurosurgical procedures or surgical procedures, the placebo is actually a sham procedure. And then there are 16 high dose. There's 16 people in the high dose group. And so patients were randomized to the treatment group or the imitation or sham surgery group. And so the study consists of a blinded 12-month core study period. So you can see on the bottom, green, someone goes through screening, making sure they're a good candidate. If they're a good candidate, they have the surgery or the sham surgery. And then they get frequent follow-ups for 1 year, during which time they're blinded. After a year, they are unblinded and have the option of crossing over into the treatment group. So today, 4 of the 6 control patients in the high dose cohort have been crossed over, and then they're followed for several more years. So the last time I talked about this, we were talking about just a minor setback. So overall, the compound was found to be well tolerated, and actually it was found to lower the mutant Huntington. So you can see that in the image on the right, that was lowered. The change in the mutant Huntington was decreased by about 50%. So that's excellent. In July of last year, there was some unexpected adverse reactions in 3 people. The study was put on hold, and in November of last year, it was determined that it was safe, everyone recovered, and so the trial has been ongoing. So what have we learned since then? So signs that it works. Total motor scores, they found that there was some improvement in both the low dose and high dose cohort with total motor score. It's important to note that the low dose cohort started first, so we have more information in that group. The total functional capacity, there was some improvement there as well. And the Unified Huntington's Disease Rating Scale, there was some small amount of improvement. And so we don't know from this data what the long-term change is going to be. These are really exploratory endpoints, but it does show that there's reason to be hopeful for this as we continue to monitor it and move forward. So the last thing I'm going to show you is just a figure that was put out by the sponsor here, and it shows the total motor score of people who receive the compound and of the controls. So the green line, those are the controls. So those are the people who received the sham surgery and did not get any medication. And you can see that it only goes to 12 months. And that's because at 12 months, they were unblinded and they get the option of switching over. What they're using as a control now is the natural history. So that is the orange dotted line. So we have a lot of information on the natural history of Huntington's disease that we can compare people to. And I just want to thank everyone for contributing to studies like Enroll HD that are observational studies, because that really makes something like this possible to use the natural history. And sometimes observational studies don't seem as exciting as the drug studies, but they're really necessary. So everyone who's participated in some of those studies has really contributed to all the research that we do. The purple line is the AMT-130 low dose, and the blue line is the AMT-130 high dose. And you can see that those are a little bit lower than the natural history. We don't have significance lines on here, but it's promising data. And so the next steps for this are they're completing the enrollment in the high dose cohort in the European clinical trial. A third cohort is ongoing in the United States. So this is an ongoing trial now here. So you can ask us more about that if you'd like. By the end of 2023, additional data should be available on the 12-month follow-up. And by early 2024, the company that's sponsoring this, Unicure or Cure, is planning on meeting with the US and EU regulators to talk about next steps and advancing the clinical development of this molecule. So I'm really glad we can end on a positive note. And I want to thank everyone for coming. And I want to thank everyone who's participated in clinical trials. Even though it doesn't always end the way we want it to end, all the information we get is hugely valuable. And so it's a very selfless and gracious gift you're giving to everyone with ADHD and their families by participating in these trials. So I'll be around for questions after Dr. Kumar's talk, but thank you all for listening. And I'd like to introduce Dr. Rajiv Kumar.